Hello, everyone, and welcome to today's webinar, Guardians of Your Science, Ask the Expert Series, Assay Design Tools to Support Your Success. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Ben Avery, Field Application Scientist, North Carolina and Virginia, Real-Time PCR and Digital PCR, Thermo Fisher Scientific. Ben, you may now begin your presentation.
Hello, everyone. Thank you for joining today's webinar entitled Assay Design Tools to Support Your Success. I'm hoping to cover a lot of content today that you'll find really beneficial to ensure successful assay design in your lab. Some of that content will be the vast catalog of predesigned TACMAN assays that Thermo Fisher offers. In addition, our custom assay design tool, which is a really beneficial tool that you can use through the web browser to order custom designed TACMAN assays for work in your laboratory, as well as ordering individual oligos. We're going to go through several case studies, potential case study scenarios where each of those tools may be beneficial to you. In addition, I'll present content in the slides today. As well, I'll move away from the slides and navigate through Thermo Fisher's website, which, because Thermo Fisher is such a large organization, the website can be challenging to navigate at times with all the products Thermo Fisher sells. So I want to make sure you're able to navigate the website efficiently as possible and find the products you need. With that, I'll start the presentation. My plan for this webinar was to hopefully present some content that's beneficial to a wide range of scientists, researchers, laboratories. So some of the individual scientists or laboratories that may find this content useful are labs that are considering using TACMAN Chemistry. Maybe you're a cyber green user, you've never used TACMAN Chemistry, and you're considering transitioning all your cyber green assays to TACMAN Assays. Maybe you're a researcher that's committed to using TACMAN Chemistry, but you're just inexperienced with designing probe-based assays, so you want some additional support designing your probe-based assays. Maybe you've searched for a pre-designed assay within our vast catalog of pre-designed TACMAN assays and can't find what you're looking for because the research you're doing is in a species or organism that isn't typically researched, or you're looking at a gene target that isn't a commonly researched gene target. Maybe you have a really aggressive timeline and you need a cookie cutter answer or a really quick solution for the technical problem you're addressing, so you need to get the materials for assay design to your lab as quickly as possible. Maybe your laboratory is very experienced with assay design, but you're interested in simplifying your workflow so that you can move from R&D into a production environment. Or maybe your laboratory is extremely comfortable with assay design, but you want to screen multiple primers and probes. You want to screen multiple oligos for finding the best design, best combinations of primers and probes possible to implement into your laboratory's workflow. So hopefully the content in this presentation will benefit all of those potential customers. Before we jump into the meat of the presentation and navigate the Thermo Fisher website to find these different products and find these different tools, I think it would be wise for us to just have a brief level set on what is TACMAN chemistry and how TACMAN chemistry works. So to fully understand TACMAN chemistry, it's important to understand the concept of FRET. FRET stands for fluorescence resonance energy transfer. And in this image over here on the left-hand side of the screen, you see an intact TACMAN probe. In an intact TACMAN probe, the reporter molecule, which is this green molecule here, is going to be in close proximity to the quencher molecule. The quencher molecule is this red molecule here. This gold connection between the reporter and the quencher, that would be your 20 to 30 base sequence-specific oligo. Whenever the reporter and quencher are in their intact conformation, meaning that the reporter and quencher are in proximity to each other, when light from the excitation source on your real-time PCR instrument, whether it be a halogen lamp, LED, argon laser, whenever that excitation source strikes the reporter dye and excites the reporter dye, energy will be transferred from the reporter to the quencher so that no fluorescent signal is emitted. And this will be the case as long as the probe is in its intact conformation. If at any point the probe is split or cleaved, as is shown in the image here on the right, when the light from the excitation source in your real-time PCR instrument strikes the reporter dye, it can no longer participate in fret. It can no longer transfer that energy to the quencher molecule, so a fluorescent signal will be emitted. To zoom out just a bit and look at how this works in the context of a real-time PCR reaction. For a real-time PCR reaction, you'll always start with some sort of template nucleic acid, whether it be DNA or cDNA. You'll start your PCR at high temperature so that you denature your nucleic acid so that it's double-stranded, or single-stranded as shown here. At high temperature at 95 degrees, your primers, shown here and here, will not have yet annealed. Your probe also will not have yet annealed. The temperature will begin to decrease, and as the temperature drops down, your primers, shown here, and probe will anneal. The probe will anneal first at around 70 degrees. The primers will anneal second at around 60 degrees. Once the primers and probe have annealed, during the extension phase of PCR, the Taq polymerase shown here will find the 3' OH of those primers and begin extending off of the 3' OH of those primers. The Taq, in addition to having polymerase activity, also has 5' nuclease activity. So the Taq has no issue, no problem chewing up and digesting the probe that's been bound to the template nucleic acid sequence. Once the Taq has digested that probe, the quencher and reporter will no longer be in proximity to each other, and a fluorescent signal will be emitted by the reporter dye. That fluorescent signal will be captured and measured by your real-time PCR instrument as shown here. So when we discuss TACMAN probes, it's important to understand that there's a few different types of TACMAN probes available for use. So the top TACMAN probe here, shown here, is what's referred to as a TAMRA probe. The quencher molecule for TAMRA probes is a fluorescent molecule called TAMRA. TAMRA, even though it fluoresces, it can still participate in fret and receive energy from the reporter molecule. These TAMRA probes, they're a bit of an older technology, and they're what are also referred to as long probes. So as mentioned on the previous slide, it's important for the probe to have an annealing temperature 10 degrees higher than the primers. And the most straightforward, kind of most classical way of doing that would be to have the probe be longer than the primers. So these TAMRA probes were, generally speaking, long probes. There's a slight disadvantage to using long probes. The disadvantage of using long probes is that they're just less specific, less discriminatory than shorter probes. So if you imagine a situation where you're looking to identify, a single base substitution, potentially in like a somatic variant, this TAMRA probe may anneal even in the presence of a single base mismatch in the nucleic acid sequence. So just know that TAMRA probes are long probes. Long probes are just less specific than shorter probes. Another type of probe that is more commonly used currently is what's referred to as an NFQ MGB probe. NFQ MGB probes have a non-fluorescent quencher molecule on the 3' end of the probe. The NFQ stands for non-fluorescent quencher. And in addition, there's an MGB, or a minor groove binder. This minor groove binder is bound to the non-fluorescent quencher, and the minor groove binder serves to increase the annealing temperature higher than it otherwise would be based on the length of the probe. The advantage of increasing that annealing temperature is that you can design a shorter probe without having to sacrifice annealing temperature. And the advantage of having that shorter probe is that it's going to be more specific. So in contrast to the TAMRA probes, these NFQ MGB probes are much shorter, more specific, and less likely to be tolerant of a single base mismatch. And then lastly, a third type of probe that is available is what's referred to as a QSY probe. These QSY probes have a QSY quencher, The QSY quencher is also a non-fluorescent quencher. And these probes are extremely useful if you're choosing to multiplex more than 2 targets. If you're choosing to multiplex more than 2 targets within a given reaction, there's a strong recommendation to only have up to 2 NFQ MGB probes within that reaction. These NFQ MGB probes have a difficult time quenching the more red dyes, such as Abby or June, whereas the QSY quencher is much better at quenching the Abby and June dyes. So if you do want to do a multiplex with 3 or more targets, including Abby and June, the QSY quencher is the best option for you. One more thing I think we should just briefly level set on is a little bit of content around the nomenclature of TACMAN assays. So if you hear me or anyone else within the Thermo Fisher organization refer to the TACMAN probes, what we're talking about is a single oligo that has a 5' reporter dye and a 3' quencher. So TACMAN probes are just a probe, just that single oligo. If you hear anyone speak of TACMAN assays are the combination of that single probe oligo with the 5' reporter and 3' quencher. And in addition, there's a forward primer and a reverse primer in either single tube format or some other format. So I hear a lot of people refer to TACMAN assays as TACMAN probes. So I think it's an important distinction to make that TACMAN probes are in fact a single oligo, whereas TACMAN assays are referring to at least 3 oligos, a forward primer, reverse primer, and a probe. And this is an important distinction because when talking about assay design, the design of the primers can sometimes be just as important as the design of the probes. When we talk about TACMAN assays in terms of, there's 2 broad categories that are worth mentioning, one of which would be gene expression assays. And gene expression assays are what I just described, where in a single tube format, there would be a single forward primer, a single reverse primer, and a single probe oligo with that 5' reporter dye and 3' quencher dye. Another type or application of TACMAN assays would be genotyping assays. In genotyping assays, there is a forward primer, a reverse primer, and then 2 different sequence-specific oligos. Those 2 different sequence-specific oligos will target specific alleles. Those 2 sequence-specific probes will have different 5' reporter dyes on them, almost always FAM and VIC, so that you can do genotyping within your lab. And then one last bit of nomenclature just to kind of speak of. When we discuss pre-designed TACMAN assays, these are essentially synonymous with like off-the-shelf TACMAN assays. So Thermo Fisher has a large catalog, over 21 million pre-designed TACMAN assays, of which Thermo Fisher has done the design and owns the designs. The other option for purchasing TACMAN assays is to purchase custom TACMAN assays. If we're speaking about custom TACMAN assays, there's a couple of different avenues to pursue there. You can purchase a custom TACMAN assay where you have done the design work yourself. So you can submit your forward primer, reverse primer, and probe designs, and Thermo Fisher can synthesize a custom TACMAN assay with your specific designs. You can additionally submit a fragment, and Thermo Fisher will do the design work for you, and you'll receive a custom assay of forward primer, reverse primer, and probe, but Thermo Fisher has done the design work for you. So both of those would encompass custom TACMAN assays, whereas the pre-designed or off-the-shelf TACMAN assays are the ones in which Thermo Fisher has the design. So in addition to those custom and pre-designed TACMAN assay options for customers, the TACMAN assays can be ordered in multiple different formats. So one of the most commonly ordered formats is the single tube assay. So these single tube assays are just what they sound like. You'll receive a single tube shipped to your lab that will contain the forward primer, reverse primer, and probe within that tube. These single tube assays are commonly shipped at a 20x concentration, so that if doing a 20 microliter qPCR reaction, you just have to add 1 microliter of this single tube assay to each of your reactions. And the single tube assays can be purchased in volumes from as low as 75 microliters, or 75 reactions, I should say, up till 2,900 reactions. If you're going to transition from the single tube assay format to more of a production operation. These pre-spotted plates, referred to as TACMAN array plates, those would be the same pre-designed or custom TACMAN assays that we briefly discussed on the previous slide, but they're going to be lyophilized in either a 96 well or 384 well plate format. This allows for higher throughput in your lab, ease of execution within the lab, really useful for production environment. But because of the configuration of these 96 or 384 well TACMAN array plates, you don't lose the flexibility to test any specific number of targets, any specific number of genes of interest that you're interested in. Another option would be our 384 well TACMAN array cards shown here. With these TACMAN array cards, there are 8 fill ports, and each of those 8 fill ports will then fill 48 different pre-spotted assays. So these are extremely useful option if you have a large number of targets to interrogate, a limited volume of sample with which to work, because each of these reactions on this TACMAN array card will be an extremely low volume reaction, but you do lose some of the flexibility that you would have with these 96 or 384 well pre-spotted TACMAN plates shown here. And then lastly, our TACMAN assays, as well as custom assays, can be ordered in the open array plate format. So the open array format can be run on our QuantStudio 12K instrument. The plate shown here has what's referred to as 48 subarrays. Each of those 48 subarrays has 64 through holes, and each of those through holes is spotted with either a pre-designed assay or a custom assay. So you can run up to 48 samples with up to 64 different targets on this specific plate. Within one experiment on the QuantStudio 12K, you can place 4 of these plates on the instrument. So you can do up to 12,000 reactions on the QuantStudio 12K at a time, which kind of led to its name of the QuantStudio 12K. And so now that we've kind of level set on what TACMAN chemistry is, some of the nomenclature of TACMAN chemistry, the different types of probes, different formats you can order assays, in which you can order assays, let's jump into the meat of this presentation and the content that I think will be really useful to listeners. So our pre-designed TACMAN assays, as mentioned, we have over 20 million pre-designed TACMAN assays available. They cover numerous different species of organism, more than 35 different species of organism. And as shown here on the right, there are assays designed for various different applications. When searching these assays online, you can filter them based on numerous different filtering or sorting criteria that I'm going to demo in just a second. And one thing that's important to know about these pre-designed TACMAN assays is that the primer and probe sequences of these pre-designed assays will not be disclosed if you purchase any of these off-the-shelf assays. However, even though you won't receive the primer and probe sequences, you'll receive a general context sequence, something that's referred to as a padded amplicon sequence, which will give you a pretty good idea of where the primers and probe are annealing to the transcript or to the genome, and it will give you the amplicon context sequence within that padded amplicon sequence. So let's look at case study 1 here. So case study 1 reads, a researcher is studying programmed cell death ligand 1, so PDL1, studying the expression levels in non-small cell lung cancer. This researcher has no experience designing probe-based qPCR assays and has an aggressive timeline for completing this project. So given case study 1, because the species of interest of here is human, because PDL1 is a pretty well-studied biomarker, I would be pretty confident there's a pre-designed assay available in Thermo Fisher's catalog that would meet this researcher's needs. So I'm going to navigate away from the slides for just a second and go to Thermo Fisher's website. And now that we're on the Thermo Fisher website, let's say we want to search for that PDL1 assay that we're interested in. So from this drop-down menu here on the left, I'm just going to select TACMAN Assays. And within the search bar, you just type in the name of your gene of interest. So I'm going to type in PDL1 and hit enter. And so as you can see, there were 245 hits or results for assays and arrays. So 29 of those results are gene expression assays, 188 of those results are SNP genotyping assays, and 28 results are copy number assays. Because case study 1 said the researcher was interested in expression levels, let's take a look at these 29 gene expression assays. So now that I'm on this screen, our goal would be to narrow down these 29 PDL1 assays to the one or ones or multiple assays that are most beneficial to the researcher. So the first filter, and your filters are shown here across the top where it says filter your results. The first filter I would always use would be species. And so because we know we're looking at non-small cell lung cancer in human specimens, we'll select human. And that's going to filter us from 29 down to 8 results. So let's look at some of the other filter options we have. So we'll start from the bottom up. So this bottom filter says does not detect genomic DNA. So if the researcher really wants to truly only amplify mRNA, if they truly want to only amplify the transcripts and not have the primers and probes anneal to genomic DNA, which is likely the case, they could select this filter. And now we're going to go from 8 results down to 6 results. So we filtered out 2. This next filter up says 3' most. So because RNA degrades from 3' to 5', some researchers may want to confirm that they're amplifying intact transcripts as opposed to just fragments of transcripts that may be remaining. So if that was of interest to the researcher, you could select this filter here. The next filter up is inventory. Inventory means that this specific assay is on the shelf and ready and in stock and able to be shipped to a customer, right away. And so because this researcher mentioned that they have a really aggressive timeline, let's filter this by only inventoried assays. And so we've now narrowed it down to 4 results. And so the last filter here under this assay attributes dropdown of filters is best coverage. Best coverage means this particular assay will cover the most different RNA isoforms, most different known splice variants for this specific transcript. Another way to kind of say that is of the transcripts in the RefSeq database, this assay will anneal and align with the most RefSeq entries in the RefSeq database. So let's just select best coverage here. And now we've narrowed it down to 1 assay. So I'll just point out a couple of other filters, but we don't need to narrow down our results any further. So this assay design filter can be really useful. So a lot of times if you want to detect mRNA, you want your probe to span an exon-exon junction. So you can specify that under this assay design filter. You may want your primers and probes within a single exon. If that's the situation, you can specify that under this assay design filter. In a situation where you're working with like a transgenic model and you want to ensure that your assay is not going to cross-react with some other species, this cross-reactivity drop-down, you can select different species, and this will confirm that our human assay here is not going to cross-react with the species that we may or may not select here. But since we've got it narrowed down to 1 result, we feel good about this assay. The first thing I'll point out about this TACMAN assay that is shown here is this assay ID. So assay IDs are an alphanumeric identifier that all predesigned TACMAN assays will have. There's always going to be a prefix that is 2 letters. That prefix that is 2 letters tells you the species of interest for the assay. So this HS stands for Homo sapien. The suffix will always be there. The suffix tells you a little bit about the design of the assay. So this M1 suffix means that the probe is spanning an exon-exon junction. So it's useful to have that information about the design of the assay. And then this numeric sequence between the prefix and the suffix describes the target of interest and where on the target of interest this specific assay will anneal. But this numeric sequence between the prefix and suffix isn't really of use to the end user or the customer. A couple of other things worth pointing out here. This specific assay can be ordered both with a FAM reporter dye and an MGB, or with a Vic reporter dye and an MGB. And so notice as I change from Vic back to FAM, this catalog number will change, but the assay ID will not change. And so the catalog number shown here corresponds to the specific dye being used in the assay and the volume of the assay that's being purchased. So these are essentially shell catalog numbers which will apply to any specific assay ID. So to be able to purchase an off-the-shelf TACMAN assay, you need to know both the assay ID you want to purchase as well as the catalog number, just with this catalog number corresponding to the dye and the size of the assay that you want to purchase. And so for this FAM MGB assay that the researcher may purchase, you can see you can purchase from as few as 75 reactions up to 2,900 reactions. A couple of other things worth pointing out on this screen is if I were to view the assay map, it will display the gene or its transcripts, and what is highlighted here in green and red is showing the assay that we've selected. And so it shows where on the genome this particular assay will anneal. So if for some reason this assay doesn't work, well, let's say we want an additional assay and you want the assay to be, just a little bit more 3' of where this specific assay anneals. You can jump to the very next assay. And so this is a different PD-L1 assay, which you can see it will span a different exon-exon junction as opposed to the one we've got selected here. So you can see there are various TACMAN assays available covering a broad range of this PD-L1 gene. So the next thing I'll point out within this screen is you can see there's a publication down here. Anytime you perform a search for a TACMAN assay and the specific TACMAN assay that you found has been used and cited in a publication, and you can see this assay ID is present right here within this publication, as well as several other assay IDs, you'll have a link on this page to the publication. And so it's really useful to see what other researchers are doing with the same assay that you may or may not purchase. So you can learn a lot about what other researchers have done with this specific assay. And then kind of lastly on this screen, I was hoping to show you, if you were to select this View Details button, it will give you some additional information and give you some links to the RefSeq database. So if I were to select this RefSeq link right here, it will navigate to the RefSeq database, and you can scroll down to the bottom of the link where it has the sequence for the transcript. And then if we'll jump back to the assay page, it's telling us that the amplicon that's going to be generated by this assay is going to be 77 base pairs long, and the assay location is location 503. And so if we jump back to RefSeq and find location 503, so location 503 would be right here, you know that the probe is going to anneal within this location. You don't necessarily know if it's the front of the probe, back of the probe, or the middle of the probe, but the probe will anneal to location 503. And since we know that we're going to generate a 77 base pair long amplicon, you can go 77 bases upstream, 77 bases downstream, and you're going to generate a sequence or a context sequence. It's going to be 154 bases in length, and that's what is referred to as a padded amplicon sequence. So that padded amplicon sequence will contain the annealing point of your primers and probe. It'll contain your true amplicon sequence generated by this assay, but you will not get the exact primer and probe sequences. And so all of that information can be obtained just publicly from these TACMAN assays without the primer and probe sequences being given to a customer that were to purchase this assay. And so if I navigate back to the presentation quickly, I think case study 1 has been pretty well addressed. So this researcher should be able to find that PD-L1 assay on the Thermo Fisher website. Since it's an inventoried assay, it should be able to be received pretty quickly, and they should be able to get moving on their project right away. And so let's imagine for a second that we were not able to find an assay that worked for us within the Thermo Fisher catalog of assays. So let's say we're doing research on a species that isn't commonly researched. If you weren't able to search within the Thermo Fisher website and find a pre-designed assay that works for you, our custom assay design tool would be a really nice option for you to use. So to use our custom assay design tool, customer would need to either paste in or type in a fragment sequence that's between 61 and 5,000 nucleotides long. The customer will then have the ability to specify a location within that sequence that they would like targeted for the assay design, if that's something they need. So if you're pasting in a fragment sequence that has a specific exon-exon junction already baked into the sequence you're pasting in, you can indicate you want to design around a specific location in that sequence. If you don't have a specific location you'd like the assay designed around, you can just select any, and the algorithm will design the assay to the best possible spot within the fragment that's been pasted in. Once the online tool has generated a design for you, you'll receive an email indicating that your custom assay design has been generated, and that custom assay design will have its own specific assay ID. So similarly to the assay ID shown on the website previously, if you design a custom assay for yourself, you'll receive a custom assay ID that corresponds to your specific assay ID, which allows you to order that custom assay. And then, when you run out of material, it makes for ease of use for reordering that assay because you already have a custom assay ID. And so one good thing to know about these custom assays is you will be able to receive the primer and probe sequences for your custom assay in most all circumstances. And I'll show you exactly when you will and will not be able to receive those primer and probe sequences when we navigate back to the website. So let's jump to case study 2. So in case study 2, a researcher is studying the expression of heat shock proteins in sea turtles. The researcher has been using Applied Biosystems cyber-green chemistry, but would like to transition to TACMAN chemistry so they can potentially multiplex in the future. This researcher has never designed a probe-based assay before. So for this specific case study, the first thing I may do, and we'll just kind of navigate back to the main Thermo Fisher splash page or the Thermo Fisher homepage. So the first thing I might do would be to just do a search of TACMAN assays. So I'm going to select TACMAN assays, heat shock protein, the target of interest for this researcher. And so typing in HSP up there, we get a lot of results. I'm again going to search the gene expression assays. Going to filter by species. And you can see there are actually a lot of different species of both plants and animals here that would be helpful to researchers. However, sea turtle, which is what this one specific researcher said he was interested in, is not present here. So we're not going to be able to find a pre-designed assay for this specific researcher. So we need to use our custom assay design tool. So just navigating back to the Thermo Fisher homepage, to find the custom assay design tool, you can navigate to this button up here that says Popular, and then from there navigate to where it says TACMAN RealTime PCR Assays. From this screen, what you want to do is just launch the custom assay design tool, which is what this button indicates right here. And now within the custom assay design tool, you can see there are two kind of main buttons here, one for ordering genotyping assays, one for ordering gene expression assays. This specific researcher asked about ordering gene expression assays, so we will click here. The first question you'll be asked when ordering this custom TACMAN assay is, what is your species of interest? And so you can choose from one of the species listed here if you like. If your species is not listed, just select not listed here. Then it'll ask if you would like bioinformatic analysis done. If you select no here, then no bioinformatic analysis will be done, and you will receive the primer and probe sequences for your TACMAN assay. If you select yes here, a proprietary bioinformatic algorithm will be used to make sure that the sequence you're interested in aligns with known transcripts for your species of interest, and it'll assess the specificity of the designs against known transcripts of your species of interest. So if you select not listed here, then this bioinformatics cannot be done. One important thing to know about if you select yes here for bioinformatics, if you select yes here because it is a proprietary algorithm to run that bioinformatics, you will not receive the primer and probe sequences. So what I see a lot of customers do is, either select not listed here or select their species of interest if it's present, and then just select no, they don't want any bioinformatics done. After you've made these two designations, you'll see this screen appear. And from this screen, the first thing you'll want to do is give your custom assay a name. So let's say we're looking at heat shock protein. Let's call it assay 1 for heat shock protein. You're going to want to then either type in or paste in a sequence that is between 61 and 5,000 nucleotides in length. And so we can briefly type in a sequence there. And then you're asked if you would like to permanently hide your primer and probe sequences. If you want to receive the primer and probe sequences, if you don't want them hidden in the documentation you'll receive with this assay, make sure you select no here. Then you can click check format. It will check the formatting of what's been pasted here and check the formatting of your naming convention here. So it says valid, so my formatting is valid. And then from there, you're given the option of manually assigning a location for the assay to target. So you can manually type in, hey, I want it to target position 35 in this fragment. Or if you don't have a specific position you want targeted, you can select automatic, and the assay will target whatever the best location is for designing primers and probes given this fragment. You hit continue. The algorithm will begin to work to design your custom assay, and once this is complete, you'll receive an email saying that the assay design is complete, and it will have the assay ID for your specific custom TACMAN assay. So I know that was a lot of content pretty quickly. I'll review it again kind of briefly, but this time instead, I'll just show you a little bit of difference. If we go back to the Thermo Fisher main page again, I'll show you how a genotyping assay would be ordered. So again, we go popular, TACMAN RealTime PCR assays. And then from there, you launch the custom assay design tool. And so previously we selected order custom gene expression assays. Now we're going to order a custom genotyping assay. We can name our genotyping assay. Let's just call it that. You can paste in or type in a nucleic acid sequence of interest. And then the only major difference for a genotyping assay is the specific SNP location that you intend to target. You have to put that in brackets and then a slash between the two potential bases or two potential SNPs at that specific locus. So given this fragment pasted in here with the brackets around these two potential SNPs, we will be able to order a custom SNP genotyping assay with one probe targeting the T sequence here and potentially being FAM labeled, the other targeting the C sequence here and being VIC labeled. And so that was a pretty quick overview of our custom assay design tool. At the end of the presentation, I'll cover all of the resources that Thermo Fisher has available for you to reach out and get assistance with walking through this. I know it's a lot to walk through and kind of a lot to digest in a short period of time. So by all means, reach out to me or my colleagues within the Thermo Fisher organization for assistance kind of going through this process. So now I'll jump back to the slides quickly. And so case study 2 should be pretty well addressed. Researcher for case study 2 should have gone to the custom assay design tool, pasted in the fragment they're interested in for their heat shock protein mRNA, and should receive a custom TACMAN assay targeting the mRNA for their heat shock protein. So let's jump ahead to a new scenario. So now, rather than ordering either pre-designed assays or custom TACMAN assays, now we want to order individual oligos. So we want to order individual primers. That can be accomplished via Thermo Fisher and through the Thermo Fisher website. These individual primers, you'll need to specify the sequence you'd like, the scale of the primer, purification method that you would like, the formulation, and then if you like any 5' or 3' modifications, which is never really used for qPCR, but may be used if you're doing, capillary electrophoresis or some other application. You can put 5' and 3' modifications on the primers. You can also order individual custom TACMAN probes from Thermo Fisher. So to order these individual probes, again, you'll need to specify the sequence of the probe that you'd like, the scale you'd like to order, the quencher molecule, the reporter molecule that you'd like on your probe, as well as the format of the probe. So to demonstrate this, let's look at case study 3. And so in case study 3, it says a lab is developing a molecular diagnostic assay to detect isocitrate dehydrogenase 1, so IDH1 mutations in patients with acute myeloid leukemia. The assay developers want to screen multiple primer and probe designs before finalizing the design of their assay. They need to order individual oligos and test different combinations of these oligos to determine which design or which combination of primers and probes is most sensitive and specific to the IDH1 mutation that they're attempting to identify. And so to assist this lab in case study 3, we'll jump back to the Thermo Fisher website. And so we're just on the main splash page on the Thermo Fisher website. To order individual oligos, again, you'll go to this button up here that says popular. And one of the options is oligos, primers, and probes. And so if you select oligos, primers, and probes, you'll be taken to this page. From this page, to order the primers, what you're going to want to select is custom DNA oligos. So if you select custom DNA oligos, you can order your oligos, in tubes or in plate format. So it sounds like the lab in case study 3 just wants to screen multiple primer designs. So I'm sure they want small volumes. I'm sure they want them in single tube format. So we'll order these in single tube format. And then you're taken to this screen. And so from this screen, the first thing you'll be prompted to do is, let me just briefly log back in here. So from this screen, the first thing you'll be prompted to do is to provide a name for your oligo. So we'll just call this IDH1, forward primer, and it's design number 1. You're required to provide a researcher name, so we'll put a name there. And then you can either, again, type in or paste in the sequence for your oligo. And then down here are different filters, different options for exactly what you want the material you receive to be like. So you can choose the scale of the primer you're ordering, however many nanomoles or micromoles of the primer you'd like to order. So we'll select 50 nanomoles. You can choose the purification you would like used for this particular primer. So you can choose to have the primer, just desalted. You can have it cartridge purified, so using reverse phase chromatography. You can have it HPLC purified, or you can have it purified via polyacrylate gel for electrophoresis. And then also you can choose to have the receive the primer either dry, meaning it's going to be lyophilized in a tube so that you reconstitute the primer when you receive it in your lab, or you can receive the primer in solution in water at some specific concentration of your choosing. And so that's a pretty straightforward, pretty simple approach for ordering primers. And then to order additional primers, you would just hit add another oligo and then paste in the information, or enter in the information here for your second forward primer for IDH1, your reverse primers for IDH1, for however many oligos, or however many primers you intend to order. So that's how you would order custom primers. We'll just go back to the Thermo Fisher main page again. So now, in addition to those individual primers that we want to order, we also want to order some individual probes. So again, we'll go to popular, select oligos, primers, and probes. And from this screen here, now rather than selecting ordering custom DNA oligos, we want to order custom TACMAN probes. And so you can see from this screen, you can select if you'd like to order an MGB probe, a TAMRA probe, or a QSY probe, or you can just click this red button, which will give you the option of ordering any of those three. And so from this screen, the first thing you'll be prompted to do when placing the order for your custom TACMAN probe is give your order a name. So it's named here, IDH Assay Development. You will be required to give your probe a name. So this would be like IDH1 probe, probe design number 1, whatever naming convention or naming nomenclature you like to use for your probe. You can paste in or type in the sequence that you like to evaluate for this specific probe. Choose the scale or the amount of the probe you'd like to receive. Choose which quencher molecule you would like to have on the probe. And what you can notice is that depending on the quencher molecule chosen here, you will have different 5' reporter dyes available for choosing. So if you select that you want an MGB NFQ quencher, you see that you can order FAM, VIC, NED, TET and NED as the reporter dye, but you don't have the ability to order ABI and June if using an NFQ MGB quencher. If we now transition to this QSY quencher, now we do have the ability to order an ABI and June probe. So just know that the quencher you choose does impact which of the 5' dyes are available for you for that specific probe. And similarly to the primers, the probe can be either received lyophilized or dried down, or it can be received in solution already reconstituted. Totally up to you how you would like to receive this probe material. And similarly to with the ordering of the primers, you can hit add oligo, and it'll give you, the room and the ability to order multiple probes kind of all in one order. And so that covers ordering custom primers and probes as individual oligos via the Thermo Fisher website. And so now I will jump back to our slides. And so case study 3 has been pretty well addressed. This lab, this molecular diagnostic lab, will be able to order the individual primers, individual probes they want to be able to screen different assay designs for kind of moving forward on this IDH1 project. So now let's say let's jump to case study 4. So now let's say that same lab, they have kind of solidified their assay designs. They know which primers and probe perform the best, and they know exactly what they want the primer and probe sequences to be moving forward. But now they're transitioning, from like an R&D environment to more of a production style environment. So they're going to need to increase the throughput within their lab. They may want to make the workflow within their lab a little more straightforward. So right now the technicians within their lab would be adding, forward primer, reverse primer, and probe to every master mix that's prepared. Maybe they would much rather have, a 20x TACMAN assay with that specific forward primer, reverse primer, and probe within it. So rather than adding 3 different oligos to every master mix, you have one assay mix, one custom designed TACMAN assay with the customer sequences in it for ease of use in the laboratory. So that is also possible via the custom assay design tool. And I'll just read case study 4 here for you, which virtually says what I just described. The assay developers from case study 3 have decided on their final primer and probe designs. They now want to order these designs in a single tube assay or a TACMAN array plate format to simplify their workflow and essentially to increase the throughput within their lab. And so if I once again jump out of the slides and navigate back to Thermo Fisher's main page, and we're actually going back to the custom assay design tool, which you've already seen. So once again, I highlight over this popular button here, select TACMAN RealTime PCR assays, and then from there, launch the custom assay design tool. And previously, when I demoed this custom assay design tool, we selected ordering custom genotyping assays or custom gene expression assays. This is where you would paste in a fragment and Thermo Fisher would do the design work for you. But in this specific case, for case study 4, where we've screened the different primer and probe designs, and it's the actual customer's design that they want in a custom TACMAN assay, you can go to this button here where it says enter custom primer probe pairs. And so if you were to select that, you're taken to this screen and you'll be prompted to enter an assay name. Similar to ordering the oligos, enter your forward primer sequence, enter your reverse primer sequence, enter your probe sequence. You can check the format again, and similarly to with the other custom TACMAN assays, after you've entered this information, you'll get an email sent to you with the assay ID for your custom designed assay, and then you'll be able to order that material using that custom assay ID. So this is the tool you would use for entering your own primer and probe sequences, for ordering your own custom TACMAN assay with your individual designs. And so if we jump back to the slides for the final time, I believe. So case study 4 should be pretty well accomplished now. You can use the custom assay design tool to generate a custom assay design that has the customer's primer and probe designs within it. So fully customizable using the custom assay design tool. And so next, just one quick designation that I think is important to point out. With ordering all these assays, whether it be an off-the-shelf assay or a custom assay, there's 2 different numbers that'll be needed for ordering the assay. One of which is referred to as the part number or catalog number. This is that shell number that I showed you that would change based on the volume of the assay being ordered, the dye of the assay being ordered, and it'll be a different part number depending on if it's a pre-designed assay or a custom assay. So that would be the part number or catalog number. The part number and catalog number have nothing to do with the actual design of the assay, what species it's targeting, what transcript it's targeting, information like that. Part number, catalog number just refers to dye, volume, scale, things like that. The assay ID, on the other hand, that's that alphanumeric sequence I showed you, where the prefix refers to the species of interest. So for the PDL1 assay we looked at, it was HS for Homo sapien. The suffix gives you some information about the assay design. So we saw a PDL1 assay that had the M1 suffix, with M1 meaning the probe spans an exon-exon junction. And then between the prefix and suffix, there was a 7 or 8 digit numerical identifier. Similarly, for the custom TACMAN assays, as I mentioned, you'll receive in your email an individual unique assay ID for your specific custom assay. So to be able to place an order for either an off-the-shelf assay or a custom assay, you need both the part number as well as the assay ID. You need both of those numbers. So the 4 case studies that I have spoken to thus far, those were hypothetical scenarios that were kind of made up and you could apply to different labs in different regions of the country or the world. The case study I'm going to highlight now is an actual true case study. So one of our inside sales representatives, Sadia Shoaib, she connected with Eva Shinkman via LinkedIn. And Eva was a researcher and a PhD candidate at Eastern Virginia Medical School. And her research was focused on specific senescence biomarkers in mouse embryos under stress conditions. And Eva had a lot of institutional knowledge, really strong background in embryology, just reproductive health, and in vitro fertilization. But Eva was not extremely familiar with real-time PCR and had very little experience with real-time PCR. However, to complete her PhD work and defend her dissertation, there were, a series of qPCR experiments that she had to complete. So Sadia and Eva and I met and discussed. We navigated the Thermo Fisher website to see if the senescence biomarkers that she was interested in were present in our catalog of assays. And in fact, all 5 of the biomarkers she was interested in did have pre-designed TACMAN assays available for her. So she was able to order those assays. In addition, she ordered a primer-limited endogenous control assay. So primer-limited assays are extremely useful if you're testing a housekeeping gene or an endogenous control that may be in very, very high abundance. So in Eva's case, she chose a housekeeping gene that was in high abundance, so it was really beneficial for her to be able to order an off-the-shelf primer-limited assay. Another challenge with Eva's research was the starting material were embryos. She had very little nucleic acid to start with. So she really needed a simple but very sensitive and effective approach to the workflow to complete these experiments. So she purchased our Invitrogen cells to CT kit, which essentially has a start to finish workflow for RT qPCR experimentation. She was able to complete the experiments needed, successfully defend her dissertation, and graduate and finish her work at Eastern Virginia Medical School. And Eva was able to do all of this work without doing any assay design herself. So she totally relied on the designs within the Thermo Fisher, Predesigned TACMAN Assay catalog. So she was new to qPCR, was able to use Taqman Chemistry, and didn't have to do any of the design work herself. And since Eva has finished her work at Eastern Virginia Medical School, she's now working at Art Lab Consulting, which is an IVF skills and training center located in Rocky Mount, North Carolina. And Art Lab Consulting is actually a business that Eva both founded and serves as the president of. And then the last thing I was hoping to mention today during this webinar is that when you as a customer go to navigate our website and kind of take this on your own and find the materials that you need for the success of your lab, find the tools on our website that you need to order those materials, it can be a bit of a challenging and daunting process. And I know I covered a lot of this content very quickly. It can be a little bit of a dense content, and just watching this webinar one time may not be enough to make it an efficient process for you to go online and order the materials. So just know that there are individuals within the Thermo Fisher organization that you should definitely reach out to for assistance. So we have a team of technical sales specialists that are located regionally. They cover numerous different products, so definitely reach out to your technical sales specialist with any questions you may have. We have a genetic analysis sales representatives also located regionally. These are the folks that may have sold you your real-time PCR instrument. They can also support your purchase of TACMAN assays. They should be able to help you navigate the website, show you the information you need to order these TACMAN assays. We also have a team of field application scientists located throughout the United States and globally. I'm a member of the team of field application scientists. So definitely reach out to your local FAS, have them kind of walk you through the website and navigate these tools for finding the appropriate assay for the work in your lab. And then in addition to all these options, if you're ever in a situation where you need immediate help, immediate technical support, definitely take advantage of the technical support phone number listed there on that slide. If you choose extension 212, it will take you through to our real-time PCR experts. And there's a really talented, bright group of individuals there waiting to take your call and help you and give you immediate assistance. So definitely reach out to all of these individuals within the Thermo Fisher organization for navigating the website and finding the tools that you need to make sure you're successful in your lab. And so with that, that concludes all the content that I was hoping to share in today's webinar. And so I believe we have time for a few questions.
Well, thank you, Ben, for your informative presentation. We'll now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box. We'll answer as many of your questions as we have time for. Our first question is, can I receive the primer and probe sequences of an off-the-shelf TACMAN assay for submission to a regulatory agency?
So thank you for that question. That's a really good question. Generally speaking, the answer would be no. We generally don't give out the sequences of the primers and probes for our pre-designed or off-the-shelf TACMAN assays. However, if it's for something like submission to a regulatory agency, there are two options available. The first of which would be to enter into an NDA, non-disclosure agreement with Thermo Fisher. And if that is done, you can receive the primer and probe sequences. The other option, if submitting to a regulatory agency, would be that Thermo Fisher can actually submit them to the regulatory agency for you or on your behalf. So you never see the primer and probe sequences, but the regulatory body will see them. So those would be the two options that are, potential avenues for getting the primer and probe sequences when they absolutely are needed. But as a general rule, the primer and probe sequences for those off-the-shelf assays are not given out.
All right, thank you. Can off-the-shelf TACMAN assays and custom TACMAN assays be GMP manufactured?
So the general answer there is yes. All custom TACMAN assays, all off-the-shelf TACMAN assays, if needed, can be manufactured in a GMP facility, which is ISO 13485 certified and is registered with the FDA. So yes, that can be accomplished if needed. One thing to just be aware of is that if the material is ordered as GMP, then the time required to actually receive the material will be longer than if you order the assays as RUO. But to answer the question, yes, all of the TACMAN assays, both off the shelf and custom TACMAN assays, can be ordered as GMP.
All right. Next question, can Thermo Fisher synthesize a control of known copy number for use with my custom TACMAN assay?
So yes, Thermo Fisher has several options for either for just positive controls in general, or if you need a standard for doing like an absolute quantitation experiment. Some of the different options within Thermo Fisher for ordering these controls would be ordering a gene art string. So gene art strings are synthetic double-stranded linearized DNA fragments that can be ordered. You could also order a gene art plasmid, which is just what it sounds like, a plasmid with a sequence of interest inserted. If a more, biologically relevant control is needed, you can order achrometrics controls. So there's several options within the Thermo Fisher family for ordering control material that would align with these, TACMAN assays.
All right, and it looks like we have time for one more question. Can TACMAN assays be ordered with reporter dyes other than FAM and VIC for deeper multiplexing?
So the answer to that is yes. So FAM is by far the most used reporter dye. I'd say VIC is probably the second most commonly used reporter dye. If you want to do deeper multiplexing, so 3 or more targets, assays can be ordered with other dyes. We have a specialty oligos group, and you would just need to email that specialty oligos group, tell them exactly what material you want, what dye you would like bound to the 5' into your probe, and specialty oligos can accommodate your request. And so I believe the email address for specialty oligos is actually in the slides in today's presentation. But yes, other dyes other than FAM and VIC can be ordered for these TACMAN assays.
All right, thank you. Do you have any final comments for our audience?
Just thank you to everyone in the audience for joining today. I really appreciate you joining the webinar. I hope you found the webinar informative. I know there was a lot of content covered, so please don't be shy about reaching out to myself or others within the Thermo Fisher organization so we can help you, navigate the website, choose between pre-designed assays or custom assays and find the tools that you need to ensure the success of your lab.
Well, thank you again, Ben, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.